I think Dr. No is going to go first and talk about his research, and then Dr. Shake will go, and then I will bring us on home. So take notes, ask questions if you have them.
All right, hi, good afternoon, everyone. My name is Dr. No. I'm new here, so the very first slide in my presentation is a little bit about myself slide. I have multiple degrees, right? The ones that are relevant are my bachelor's degrees in chemistry and biology, my master's degree in chemistry, and my biochemistry PhD. So by training, I'm actually a biochemist, right? Formally trained as a biochemist. And I'm also, a member of multiple different organizations that do forensic sciences, because I was a forensic science for about 5 years before I became a professor here. And then before that, I had more than 10 years of experience in biochemistry research. So biochemistry and bioanalytical. So today, my disciplines, my forensic discipline specialty is analytical chemistry and firearms. That's what they hired me at the Alabama Department of Forensic Sciences to do, to investigate crimes against persons that involve firearms. And contrary to popular belief, there's a lot of chemistry involved in that. So this is just a very funny picture that a meme that my colleagues actually put up for me the first time I ever testified in court. And when I was there, the judge didn't believe that I was actually a forensic scientist, because apparently I looked kind of young, and this is before COVID too, so I, no mask either. And he's like, "You're not Dr. No, you're not a forensic scientist," like not believing who I was. And I'm like, "I am, Your Honor, I'm Dr. No with the Alabama Department of Forensic Sciences." And it got to the point where he just didn't believe me so much that he had the bailiffs come in to take me away, holding me in contempt of court, right? Luckily, the Attorney General of Alabama was actually there, and he finally heard what was going on, and he stood up and was like, wait, that's Dr. No. Don't arrest him, he's my witness. So before I even got back to the office, I guess word spread pretty fast, and these pictures of this were all over the office walls in my office, had already been printed out all over the walls, and they say, "Honest, Your Honor, I am a forensic scientist." So word got out pretty fast. I tell you the story because it's kind of like the same way I kind of run my lab. It's like, it's going to be a fun thing to do, right? But at the same time, you're going to learn a lot of practical applications, right? So if you ever go to court and you get held in contempt, I'll make a picture of you and do this kind of thing around our lab. Anyway, so things to keep track of, one thing I need to keep track of is the time. But some things to keep track of, keep in mind, is that forensic labs are not what you see on TV, right? They're not like CSI New York. They're not like CSI Miami, where there's like multiple screens or touch screens that are the size of 55-inch TVs. There's not these things that are like holograms. Forensic labs are not like that. And the point of me saying that is because forensic labs, vast majority of them, the government ones, right, are underfunded and they're underemployed. Like there's not enough people working there, and there's not enough money to buy equipment, right? And so when you do any kind of research in forensic sciences, you have to keep these things in mind, right? You have to remember, again, there's a lack of funding for the purchase of new expensive instruments. There's not enough time for the scientists there that are working to even get trained, because most forensic scientists are already inundated with backlog cases, right? So if you want to train someone in a new forensic technique, you have to take them off of that the casework to get them trained. But DAs don't like that, judges don't like that, right? Because they need to get these cases out. So that's another challenge you have to deal with too. And then in the same vein as that, you have to train them in new chemicals too, that you might have to use in these new techniques. That's a problem as well. So every single forensic science technique that you try to implement, that you come up with a new method, which let me just tell you, the community is very resistant toward it, right? You have to address these challenges by two things. You have to utilize the existing instruments that are already in forensic labs. That way you don't have to retrain people on how to use the instruments. And then you also have to use existing chemicals, right? And it's actually a really tough thing to deal with. It's a very tough thing. Most biochemical labs, biochemistry labs, like research labs, they have the funding to buy it. But government labs, it's a problem, right? I wish we had this kind of hologram stuff. So one of the projects I'm working on right now is called quantitative distance determination. And so I actually started this project back when I was at the Alabama Department of Forensic Sciences. And what it is that you can determine how far a firearm was from a victim when they're shot, right, using the chemical traces that are left behind by that firearm known as GSR, gunshot residue, right? You might be asking yourself, why is it important for you to be able to figure out how far a gun was from a victim? Well, the vast majority of times when a victim is shot and they're dead, the suspect will always say, oh, they committed suicide, or oh, there was a struggle, or something like that, right? But if you can use chemistry to figure out how far the gun was, you can imagine if someone's holding a gun to their head, right? It's really hard to pull that gun any further than like a couple inches, right? And so if your chemical analysis shows that it's really far away, that person obviously did not commit suicide. Does that make sense, right? And then the other thing is they're like, oh, there was a struggle. Well, usually when there's a struggle, guns are really close, right? Again, if the chemical analysis shows that it was far away, then there wasn't a struggle. You shot that guy while he was unarmed or whatever. Does that make sense? So you can disprove stories that suspects tell you, right, using chemical analysis. And so there are a few different ways that they do it conventionally now, right? And this is the chemistry part of everything, right? One of them is called sodium rhodisnate. Sodium rhodisnate is this molecule right here. And long story short, when it comes in contact with lead, which is a main component of gunshot residue, right, it turns purple. It turns dark purple, right? But see, lead alone can't tell you that that it was a gunshot wound or there was gunpowder there. You have to, detect other things too. So most bullets, right, are actually covered in a copper jacket, and so you can use another molecule called dithioxamide, right? And what it can detect is it will complex copper and turn green, right? So you have a combination of purple for the lead, from the GSR, and then the green for the copper from the copper jacket of a bullet, right? That's still not enough. A couple more things have to be found. Diphenylamine, right, will actually complex the nitrate ion, which is the main component of gunpowder, and it will turn blue, right? And then nitrite, which is a component of burned gunpowder. By the way, when gunpowder is exploded, right, when it catches on fire, there's an oxidation reaction that occurs, and nitrate becomes nitrite, so you have to detect both nitrate and nitrite, right? And so the nitrite ion combined with alpha naphthol and a couple of other things will actually turn yellow-orange, right? So at a scene, we will use all four of these chemicals to figure out whether or not a gunshot actually happened, right? Because you can get a knife wound and then look like a gunshot wound. Or moths will actually eat, holes in the clothing that look like a gunshot wound. So that's important. The thing about it though is that right now, right, to figure out how far a gun is from a victim, we use visual comparison, okay? We use visual comparison. And so you can see when a gun is, by the way, no one ever gets shot with, this is a case I have to work, and no one ever gets shot with a white shirt on, right? It's always with a dark shirt. So you can never see the gunpowder, so that's why you have to do chemical analysis on it, right? And so you can see that this is the evidence hole right here. This is a chemical representation of the lead that's present at the hole, and this is the chemical representation of the nitrates at the hole, right? So what we do is we'll take the firearm that we think was used in the crime, and we'll test fire it at known distances, right, to get patterns of gunshot residue, right? This is just the normal pattern of gunpowder and gunshot residue right here, and these are the chemical analysis of lead and then nitrates. And you can see at 6 inches, it's pretty dense, and then at 14 inches, it's pretty, not dense, right? And you look at the evidence there, right? It looks like it's kind of in between 6 and 8 inches. But that's the thing. The vast majority of the analysis that we do today in this kind of distance determination is we think it's between this distance and this distance. It's a range, right? But the problem is right now, there's a huge switch to turning everything from being subjective, where we're like, oh, I think it's like this, and I think it's like this, to actually having quantitative numbers, right? And so my idea was to actually extract out all the chemicals and do a quantitative analysis on it using a calibration curve, right? So basically turning these test fires, right, extracting out the lead, extracting out the nitrates, right, and then doing a chemical analysis on it so that you can do a calibration curve of the concentration of each of those known test fires, right, and then compare it to the concentration of the evidence. So So like I was saying before, we have all these issues that we have to deal with. Number one, dark clothing. Number two, like sometimes the transfers to make the transfer the gunpowder from the dark clothing to the light clothing doesn't work out well, right? We have this problem with blood too. Blood always covers up gunpowder. What do we do with blood, right? And then again, it's very subjective. So here's the approach, right? What we can do is that we can do, like I was saying, make a calibration curve of the known distances, right, by extracting out the chemicals that we want to analyze and see if there's a correlation between concentration of the chemical, right, and the distance of the chemical, or sorry, the distance of the test pattern. Does that make sense, right? And so I've actually done a lot of this already before, right? I've figured out that the best combination of chemicals to actually extract gunpowder residue is a combination of hydrogen peroxide and acetic acid, right? Very good combination. And I've extracted a whole bunch already out. And the thing I actually used, right, was inductively coupled plasma. Has anyone ever heard of inductively coupled plasma before? It specializes in detecting metals, is what it does, right? Basically, a metal will get flamed in the plasma, and it will give off a certain spectrum, and you can identify that metal. Does that make sense, right? And so I did this, and I did it no test distances, and I got some data, right? But the problem with that is that, man, this thing is expensive. Inductively coupled plasma, specifically this is ICP OES, optical emission spectroscopy. The instrument's kind of expensive, and the crime lab's not going to buy that, right? And so I want to take what I did and move it so we can actually just use a UV vis spectrometer, right? Where most people have a UV vis spectrometer. We have like 3 of them here, right? So here's some of the data I got, right? Just real quickly. We found here that the Bashinsky transfer is how we transfer the gunpowder residue from the dark clothing to the light clothing. There's actually only about a 50% like efficiency, right? I'm sorry, there we go. There's only like 50% efficiency based on the caliber, right? Here's 9mm. That's how much ppm of lead I had there, right? But then look how much I actually transferred. So like it's really an inefficient method using Bashinsky transfer, right? Wouldn't it be cooler if we were just able to extract all that lead out and put it into a little vial and do a test on it instead of just looking at it, you know? It's a really inefficient method. So this is just a graph showing how the composition of gunpowder in metal, right? And you can see there's a lot of copper there from the bullet, right? And then barium, antimony. All those are things that people actually look for right now as far as gunpowder goes, gunpowder residue goes, along with a couple other things. But the thing I really want to show you is this, right? Notice here that at 5 inches, 10 inches, 15 inches, and 20 inches, and 25 inches, right? The concentration of each of these metals actually goes down, like in a predictable way. And I mean, this looks really flat right here, right? But that's because it's in reference to the rest of these. But I mean, look at that R squared value. I mean, that's awesome, right? If we can use that as a calibration curve to determine the distance there is, the distance of gunshot residue patterns, that would be amazing. So I used this I did this using ICP, and again, we don't have an ICP here, and most labs don't have an ICP, right? So what we want to try to do is figure out how to make this into a UV vis type of analysis, right? Which is actually kind of easy because you saw when we did the analysis with the chemicals, they had very specific colors, right? Purple, yellow, green, and blue, right? And what is the UV vis the best at? Detecting colors, right? And so it'd be super interesting if we'd be able to transfer that kind of data from the ICP to the UV vis. Okay, so that's the first project I'm working on. I started that at ADFS, got overloaded with casework, so I had to stop working on it. So I'm glad I'm going to be able to continue it here, right? And then the last one I'm talking about is called molecular cloning of STRs, right? This is kind of like it's like a pipe dream of mine, and I don't really know what the utility of it is, right? But it's kind of a fun thing, kind of crazy, one of my crazier ideas, right? What is the STR? Well, STR stands for short tandem repeats, and it is the predominant method for forensic scientists to identify a human being using their DNA, right? And it's the DNA fingerprint, right? So if you look at a DNA double helix in eukaryotes, right, different from prokaryotes, our DNA actually has in it exons and introns, right? Exons are where all the genes are that actually code for your proteins, your enzymes, all the stuff that makes you work, right? But then in eukaryotes, we have this stuff called introns, which is extraneous DNA that doesn't code for anything at all. But it's useful because in these introns we have these things called STRs, short tandem repeats, right? So these repeats right here, ACC, ACC. So the repeat is ACC, and then the thing is how many times does it repeat? For every single person, it's different, right? So you can use that as a way to identify or individualize a person, okay? So the FBI requires that we identify 13 of these loci before we can actually make an individualization. And if you have 13 and you do statistics on it, right, you can say this random person has a 1 in 608 quintillion chance of matching this DNA profile, which is more than the entire population of the world, right? And so STR analysis is quite powerful in individualization of people, right? So this is just the process right here. We take some blood cells, we do some PCR, preliminary chain reaction. I'll talk about that a little bit later. And then you can either do gel electrophoresis, which a lot of people in biochemistry know what gel electrophoresis is, right? Or do capillary electrophoresis. I'll talk about that in a second too. Well, then you get a DNA profile, right? So when you look at a DNA profile, right, this is what a normal DNA profile looks like. And I just want to point out this right here. This locus is called amylogenin right here, right? And if you have 2 peaks at the amylogenin, right, it means that you have a male. The contributor is a male. And because one of those peaks is for the X chromosome and the other peak is for the Y chromosome. So let me ask you a question to the students here, right? What if you only had 1 peak there? What would it be? Why would it be a female? Exactly, 2 X's, right? And that's why you only have 1 peak there. So this is a very fast way of determining whether or not an assailant or suspect is a male or a female, right? 1 peak or 2 peaks, right? And then the rest of it is what we use to actually identify people, right? And you can see that this is, a lot of people don't know what a chromatogram is, but that's okay. The point is that these peaks tell you how long a piece of DNA is, right? And that will translate to how many repeats are. So if you look at this guy right here, he's got AG, AT, and AGAT, 7 repeats here, 6 repeats there. Does anybody know why we have 2 of them that are the same?
One from the mother, one from the father.
Exactly, right? You have a contributor from your mom and one from your dad, right? And so this is from the mom side, this is from the dad side, right? And so your mom, I forgot which one was it. Oh, well. So either your mom or dad donated to you 7 repeats, and your mom or your dad donated to you 6 repeats, right? But everybody has a mom and a dad, and each one donates those repeats, right? So for this woman here, 4 repeats from one of her parents, and 19 from the other parents. Here, this man, 12 repeats or 14 from each one, right? And so you can even see how the statistics start multiplying on how you can individualize a person, right? You go beyond the population of the world, which I can't remember what the population of the world is, like 30 billion or something like that. I'll just tell you that 608 quintillion is huge, much bigger, orders and orders of magnitude bigger than the population of the world. So what is my question? My question is it possible to reproduce an identical human STR profile from molecularly cloned bacterial cells? Right? So I want to be able to see if I can put an STR profile of a person into a bacteria cell. And really the only practical application of this is to frame someone else for a crime, right? But if we can do that, that would be a huge thing because DNA is the gold standard right now in forensics, right? And they use that for everything. If you go to court and you don't have DNA evidence, right? You've lost the jury, right? And so it's so held so high, I want to poke some holes in it, right? And so this is one of the ways to poke those holes, because if you can figure out how to put this STR profile of anybody, say someone you saw drinking coffee at Starbucks, right, and you want to take the trash of the cup he threw away, you can put his profile, or I want to see you can put his profile into a bacterial cell. And then it has the added benefit of the fact that bacteria will actually glow under UV light just the same way that sperm will and blood will. So if you're a crime scene investigator and you see samples of biology on the floor, you're like, oh, that's a sample that just lost, right? Not even knowing that it's bacteria. You know, it could be saliva, it could be sperm, it could be blood, whatever, right? But they're going to swab it anyway. So these are the 13 plus 2 more of the loci that the FBI requires us to use to actually individualize a person, right? And you can see they all have different repeats, right? And they all have a different number of repeats, right? And so if you use all these together, you can do a really good job at individualizing somebody. So this is PCR. I'm not going to go through this real deeply, but all you really do, all PCR is it's a way to amplify a short strand of DNA, right? And make tons of copies of it. What you do with it afterwards is your choice, right? But what I want to do afterwards is actually take the primers from the PCR, right, and be able to actually amplify out an STR, right? And then after I get amplify out the STR, we have to go through and actually use gel electrophoresis to make sure we have the right size STR, right? But then this is the part that's fun, okay? We're going to try to ligate it into an expression vector. My favorite one is PetNuet, right? It has two multiple cloning sites on it, right? This dark blue one here and this green one here. And we want to be able to put the STRs from some individual, let's say me right now, right, into these sites right here. And then we'll transform this plasmid right here into a bacterial cell and let it propagate, right? Make tons of copies, right, of this one genetic profile, right, which is great because now we can use it to frame someone for murder. I'm just kidding. We can just use it as a proof of concept, right? And so the reason why we want to put it into a plasmid is because plasmids are actually independent of the genome of bacteria, right? And not only that, they are self-replicating and self-expressing. So you can actually make a ton of copies of this, right, and then have a vat or a flask of just cells, of bacteria cells, and do what we call plasmid extraction and only extract out the plasmid and not even have to worry about the genomic DNA of the bacteria that might get in the way, right? So we'll do that, and then we'll do, so I wish we had one of these, but these are probably cost prohibitive, but this is an ABI 3500 genetic analyzer. Most crime labs have one of these, right? We'll take our bacterial cell, and then we'll go into, take it here and make an STR profile out of it and see if it matches the original, right? The original that we had before. From whoever is the donator. I guess I'll be the donator first, but maybe my students could, one of you guys want to be the donator to be the one I framed for a crime, right? So anyway, that ends the presentation. So I just want to, before I go, talk about the kind of things that you'll be doing, like the instruments you'll be using, or you'll learn how to use, right? So in the firearms one, you'll definitely be using some HPLC, right? High performance liquid chromatography, or some other people call it, I only call it high performance liquid chromatography, so I'll just keep it that way. And then you'll also be using a lot of UV vis and mass spectrometry in that first one, in the firearms one. And then in the second one will be most of the biochemistry stuff, right? So you'll be using a lot of PCR, which are thermal cyclers. You'll be doing a lot of RT-PCR, which is real-time PCR. Lots of gel electrophoresis, lots of sequencing, right? Lots of ligations, and basically molecular cloning. So the basic skills that you need to work in any kind of biochemistry lab you get from doing that last project, right? Which is all the conventional molecular biology techniques: PCR, ligation, transformation, and then cell proliferation, and then DNA extraction, right? And that's it. You guys have any questions? Sorry, I get really excited about my stuff. So I get really excited about my research, and so it's kind of crazy. So anyway, any questions, anybody? Nope. Cool. All right. Thank you.